All right, everyone, welcome back. It is the Water, Wetlands, and Watershed Seminar, Fall 2020. It is October 28th, and today we have Betty Staugler is going to speak to us about issues of water quality and community sort of participation in Southwest Florida. But before we get started, anyone have any announcements about upcoming activities or events? Dr. Clark is not here. He usually announces something. Well, I will announce that when we meet again, it will be after Election Day, which means your opportunity to vote, if you haven't done so, comes between now and Tuesday. If you're in Alachua County, early vote goes through Saturday, and then, of course, Election Day on Tuesday. If you have any questions about how to vote, where to vote, you can email me, and I will hook you up with that information. Okay, so now we have our speaker for the week, Betty Staugler. Betty has her MS in environmental science with a focus on marine and ecological sciences from Florida Gulf Coast University. She's been a faculty member at through UF and IFAS Extension and Florida Sea Grant since 2004. She currently serves as the Florida Sea Grant agent in Charlotte County down in Southwest Florida and liaison to NOAA's National Center for Coastal and Ocean Science Harmful Algal Bloom Forecasting Branch. As a Sea Grant agent, Betty works developing comprehensive marine and natural resource educational programs on sustainable coastal communities and habitat and water quality issues. So she really links together a lot of what we've talked about in seminar, from the sort of biophysical side all the way to the ecological side to the human community side, this focus on habitat and water quality. As a liaison to NOAA, she coordinates the knowledge and use of research products, tools, and techniques to address harmful algal blooms in the Gulf of Mexico. So Betty, with that, I'll give you the floor and you can go ahead and share your screen.
Great. Is my screen shared? Can you not see it? Okay, hang tight. Here we go. This one.
Perfect, we can see it. Excellent.
Okay. All right. So thank you, David, for that nice introduction. And I have to say, I'm really excited to be here. Looks like you've had a great lineup of talks already, and you have a lot more great lineup coming after me. And as I was thinking about this presentation and looking at the talks that people have already presented, it made me think that perhaps I'm the only speaker in this session that does not have a research appointment. And so my faculty appointment is 100% extension. And when I talk about that with other people, even within the university system, I find a lot of times people don't really know what that means. So my next couple slides are going to give you a real quick fast track of what it is to be an extension faculty member at the University of Florida. So the University of Florida is one of 2 land grant institutions in the state of Florida. And what that means is it's the people's university. It was created or established via a federal act called the Macroalgae Act in 1862. And that act provided land that could be sold, but the proceeds had to be used to establish a people's college, which would include practical studies that were important to the people of Florida at that time. And so primarily we're talking about agriculture and mechanical science. And this was kind of in response to the Industrial Revolution and making sure that communities were not being left behind. Another important act was the Hatch Act of 1887, and that established field stations. And so it really took research and education out to the communities where commodities were being grown. And then the final act was the Smith-Lever Act of 1914, and that's the act that created cooperative extension. And so that established a partnership with local governments, and it allowed for university faculty to be set up in local communities to become trusted members of that community and serve as a bridge between the university and that local community. So when we think about extension, oftentimes this is what people have in their mind, is somebody standing up in front of a group, transferring what we know from the university out into that local community. And certainly I do that. That is me standing in the front of the room. This is a fishing club and we're talking about snook biology. But extension is a two-way street. And so oftentimes issues arise at the local level, and it's my job to align those needs with university expertise towards finding solutions. So that means I do a lot of grassroots problem solving, and that lends itself really well to citizen science. And I have to say that's one of the passionate parts about my job, is working with citizens to help them solve issues. And so with that, I'm going to lead into what I really want to talk about, which is this project. And it's called Eyes on Seagrass. So it does have something to do with seagrass. And just to orient you to where this project is happening, the little map in the bottom left-hand corner, I've circled Charlotte Harbor, which is located in Southwest Florida. Charlotte Harbor is Florida's second largest open water estuary. It is just slightly smaller than Tampa Bay to the north, but what's very different about it is that it is relatively undeveloped. And it has a very large watershed relative to the estuary size. The other thing that's kind of unique to Charlotte Harbor is that 80% of the shoreline is in permanent preservation as part of the Charlotte Harbor Preserve State Park. And this land was purchased to create a buffer between what was happening in the uplands in order to protect the estuary, which is a state designated aquatic preserve. Some other things I want to orient you to, there's 3 rivers that flow into Charlotte Harbor. We have the Peace River to the north. Let me see if I can find my pointers here. Okay, so there's the Peace River to the north, and then the Myakka River to the north. Both of these have fairly large watersheds relative to the estuary. So this upper portion of Charlotte Harbor tends to be very brown with tannins. And then in the southern portion of the estuary, we have the Caloosahatchee River, which is a very altered system that flows almost directly into the Gulf of Mexico. So we don't see that dark influence from the freshwater tannins down here. My project is going to occur up in this area of the estuary here. The last thing that I want to mention is the flow pattern of water in the estuary. So these freshwater systems, they tend to discharge down this western wall of Charlotte Harbor. And then the salt water comes in through Boca Grande Pass and up the eastern side of the harbor. And you see this really prominently in salinity data, particularly in the summer months. All right. So here, we're looking at seagrass acreage for Charlotte Harbor. This data is provided by the Southwest Florida Water Management District. And every 2 years since 1992, the district has consistently flown low aircraft and taken aerial photography for estimating seagrass acreage. And they do this generally in the winter months when the rains have stopped, so you have better water clarity. And they only fly when there's low cloud cover and significant or good enough water clarity. And so what you can see here is there's a little bit of variability in the seagrass acreage estimates up through about 2010. And a lot of that has to do with whether we're having a wet year or a dry year. But then when you get over towards 2012, 2014, 16, it looks really happy for seagrass in Charlotte Harbor. However, if I take this data out to some of my stakeholders, particularly recreational fishing interests, the guides and the anglers, they don't buy this data. In fact, they're particularly concerned about macroalgae. And they've noticed in recent years that they're seeing a lot more macroalgae in the estuary. And they're concerned that the macroalgae is being is being reflected in the SEAGRASS data. They're also particularly concerned that these increasing abundances of macroalgae are damaging the seagrass habitats and their associated economic value by outcompeting for light and nutrients, by creating hypoxic conditions, and by interfering with boating, fishing, and tourism. And what we don't know, what I don't know, is if what they're seeing is ephemeral macroalgae abundance that dissipates over time, or whether this is something bigger that we need to be concerned about. Some of these anglers spend hundreds of days on the water. If they're a professional fishing guide, they're out there a lot. So this has become kind of a real concern in my area. Now, what we know is that macroalgae are biologically important. They photosynthesize, so they provide this important oxygen source for coastal systems, and they play a really important role in ecosystem health. And this is especially true in the winter months when seagrasses are dormant and not growing. In Florida, macroalgae often display marked seasonal abundance patterns. So they peak in the spring, and then they decrease in the summer. And this kind of correlates with when the seagrasses start to grow. And we don't know if these patterns are related to that or if they're related to salinity, temperature, and or light conditions. Okay. So what we do know is that macroalgae can cause ecological concern when they grow really rapidly and get out of control. And this is being reported in many estuaries worldwide where macroalgae abundance has increased in recent decades due to nutrient over-enrichment. And studies have shown that seagrasses typically dominate in waters where there's low nutrients and high light availability, where in contrast, macroalgae typically dominate where there's less light and more nutrients. So one could expect as nutrients increase that seagrass, which are slow growing, to be replaced by macroalgae, which are fast growing. And this is kind of shown in this graphic from the Lamar Coastal Lagoon, which is in Spain. And it shows this eutrophication process that's occurred over a roughly 30-year period. So you have this really nice healthy seagrass bed, nice light availability, low nutrients. And then as we start creeping up in nutrients, we get less light and we start seeing this shift to the algal community. So more macroalgae, more epiphytic growth or plant fouling the seagrass blades, more phytoplankton in the water column. And as the nutrients continue to be loaded into the system, we have less light to where eventually we start losing the seagrass and those associated epiphytes for macroalgae and phytoplankton. And if this continues to trend, then eventually the phytoplankton outcompete the macroalgae and all you have is the phytoplankton in the water. And then it really starts showing up in the water bottle because it's no longer tied up in these plant communities. And from an ecosystem perspective, this shift from seagrass to phytoplankton could have cascading impacts as animals that are dependent on seagrass for food and shelter, think of spotted sea trout, for instance, are replaced by less desirable species such as jellyfish. All right, so what you're looking at here is a map of submonitoring that is done by the Charlotte Harbor Aquatic Preserves. They have been doing permanent seagrass transects in Charlotte Harbor since 1999. So they have a pretty robust data set. They do one survey a year, transects from shore out to the deep edge of the seagrass bed. Their sampling occurs in the fall, so it's designed to correspond with the peak of the seagrass growing season. And what they have noted over time is that they are seeing some increases in green filamentous macroalgae since about 2012. And although they don't quantify those impacts, they do make note of them in their data set. So what I wanted to do was develop a monitoring program that could complement what the Aquatic Reserves was collecting, but focus my efforts on when macroalgae, it should be at its growing abundance, so in the spring. And so to do this, I put together a citizen science monitoring program, which was piloted in 2019. And the goal of my project is to improve our understanding of macroalgal bloom dynamics in shallow seagrass areas. So I put together methods, again, that complemented but not replicated the Aquatic Reserve seagrass transect information, thinking about the type of monitoring that citizen science could do and could do well. We field tested the methods actually with the Aquatic Reserve staff, and then I began recruiting volunteers in March of 2019. The project kicked off with a training session in early April, and then immediately after, the volunteers did their sites. So they, within days, they were doing the sites that they had agreed to do. So in terms of the volunteer commitment, the volunteers were required to provide their own teams of 3 individuals. They had to provide their own boat. And remember, Charlotte Harbor, 80% of the shoreline is in permanent preservation. So you're not getting anywhere without a boat. They had to provide their own snorkeling gear, attend the training session. And then we survey twice. We survey in April and we survey in July, because I want them to see that change in seagrass and macroalgal abundance so that we can see if the seagrasses are responding in, favorably, or if we're continuing to see macroalgae over time. And I provide a 2-week window for them to do their survey sites. And so it only takes 1 day to do the surveys, but they have this window that allows for them to plan around schedules and also bad weather. They agreed to return their data sheet and a macroalgae sample, and I'll get to that in a minute, take care of their gear, and then educate others on the importance of healthy seagrass. And this last point is really important because a lot of studies have shown that participating in citizen science data collection, volunteers have a greater understanding of the subject matter that they're evaluating. They trust that data and the findings that the data produces. And as a result, they often become ambassadors of that information, and they share it within their networks, whether that be their friends, their family, clubs, or associations. So there's a lot of considerations when you're dealing with volunteers, thinking about the time of year. You know, we have to be respectful of their time. These are volunteers. If I asked them to go out and sample in February, I probably wouldn't have any volunteers because the water is cold and nobody wants to be out there. We also have to think about what it is we want to know versus what it is we need to know, because we want to limit the field activities so that what they do, they're able to do really well, so that we're able to get some meaningful data out of the volunteers. And with that, I have to make sure that the methods are appropriate for volunteers, that they are understandable, they're clear, and that leads to a lot of training and volunteer resources. So making sure that I'm spending the time with the volunteers, going over the methods. We do a PowerPoint presentation, the importance of seagrass, macroalgae communities. We go over the field methods via PowerPoint, and then we do hands-on actual training exercises. From there, the volunteers self-select their sites, and they leave with resources. So very detailed methods, as well as these this type of field resources that I put together for them that helps them identify seagrass, macroalgal color phylums. We have some brown blanket abundance scales in there that they can use to help them determine abundance estimates and other things. All right, so in terms of the study design, this is what it looks like. Again, we're focusing on Upper Charlotte Harbor right now, but we plan to expand westward soon. But the sites are randomly selected, and I did this to remove sampling bias. When you're working with volunteers, oftentimes they may want to, if you give them the flexibility and they think you want to know where seagrass looks good, they're going to go where seagrass looks good. If they think you want to know where macroalgae is really bad, that they'll go there. So we took that away by doing random points. These were generated using the FWRI sampling grid, which randomly selects 1 nautical mile grids. So from that, we can get our degrees in minutes, and then we used a random number generator to get our decimal minutes. And that generated these points. Because they were randomly selected, we knew that some points would fall on land or out in deep water. And so the volunteers were instructed, if the point was on land, if it was in too deep of water, which we called anything over 2 meters, if it was in a channel, or if it was on a sandbar or any other location which would make it unsafe to conduct a survey, they had very detailed instructions on how to get to a suitable sampling location. And from there, they deployed a 50-meter transect perpendicular to shore, and that transect was marked off at 0, 50, and every 10-meter increment. And they laid a half-meter quadrat every 10 meters, and that was what they conducted their survey in. So 6 points total on each transect. All right, so this is a portion of the monitoring gear. After I took the photo, I realized I forgot a few things like the dive flag and the cooler. But my goal when I'm doing these projects is to make sure that the volunteers have all of the resources that they need to do the survey. The monitoring gear also was constructed by volunteers. So yes, I know that quadrat is the lines in it are not done correctly. This is for demonstration purposes only. We had to restring quite a few of them to make sure that everybody had correctly strung quadrats. But the transect line is what you see in the bucket. We use pre-measured line. It's sink line. It's weighted at the 0 and 50 meter mark with dive weights. And then additional line has floats attached to it so that the volunteers can see it at the surface. And then I use duct tape every 10 meters to mark the stations in between. And it coils up really nice in a 5-gallon bucket. Other important things for the volunteers, a depth pole, which they also use to measure seagrass blade lengths. Here's the quadrat, a salad spinner for their macroalgae sample, a spring scale, and a Secchi disc. Okay, so the data that's collected, at every station, the volunteers collect at every quadrat, the volunteers collect water depth in centimeters. They determine the sediment in terms of grain size, so everything from muddy muck to shelly sand. They estimate macroalgae abundance in terms of percent cover. And then they remove the macroalgae off the quadrat so that they can expose the seagrass underneath, and they do seagrass percent abundance. And then if there's more than one species present of seagrass, they do a percent contribution to the total abundance. They then look at the seagrass blades and they give a general description of the epibionts or the fouling communities attached to the seagrass blades. And here the categories are none, light, moderate, and heavy. We don't separate in terms of plant or animal, but we will be moving forward. And then finally, for each seagrass species present, they take 3 random blade lengths. All right, so that's at every quadrat. At the 20-meter quadrat only, instead of moving the macroalgae off the site, they actually collect it in a bucket and bring it back to the boat. And with this sample, they're spinning it down for 60 seconds in their salad spinner, which I always call a salad shooter. I think it sounds so much cooler. And then they separate the algae by color phylum, and they have these field resources here to help them determine different color phylums. They remove anything that's not macroalgae, and frequently we get snails, barnacles, seagrass blades that they have to remove. And then they put each color phylum in a Ziploc bag, and they weigh it using their spring scale and then store that in a cooler on ice that they bring back to me. And what I try to do with their sample is I try to identify the dominant macroalgae, at least to the genus level, so that we can see what's out there and also estimate the % contribution of that dominant macroalgae to the total community found within that quadrat. The other important thing about having the volunteers bring the macroalgae sample back to me is it allows me to make sure that they're correctly classifying by color phylum, which they do a fantastic job of. So this is some of the data, and this was actually put together by Ashley Smith from the Soil and Water Sciences Department. Ashley is my co-conspirator on this project, and she used this in part as a teaching tool in with some of her students. So the data is divided into 3 estuary segments. This first segment here is on the eastern shoreline. So E1 is the northern, down to E3, which is at the southern end, and part of it got cut off. The U1 and 2 is up here, and then the W3 and 4 is along the western shoreline. And not a lot of surprising information here. 3 species present. We don't see Syringodium because it's much too fresh in the upper harbor. So the species that we did see present are what we would expect to see in the upper portion of Charlotte Harbor. I will point out that this team here, they did not find any seagrass in the spring. They were actually sampling in pretty deep water, and in the summer it was so deep they had to move the site. So this one we always take with a bit of a grain of salt. But what we did find that was interesting was when we looked at the macroalgae coverage in relation to the seagrass coverage. And so here, what you see on the eastern shoreline over here is a lot of macroalgae, and particularly a lot of macroalgae relative to seagrass. We did not see that in the upper harbor or on the western side of the harbor, which was surprising because this is where the Aquatic Reserves was noting macroalgae in their samples. And you notice that between spring and summer, we were not seeing that macroalgae disappear, although we were seeing some growth in seagrass as we would expect. And then here's our epibiont density. And what was interesting here was that we had less epibiont density in the summer compared to the spring in that area where we had so much macroalgae. Whereas in the other sampling locations, the epibionts grew during the summer months, which you would expect because you have more fresh water coming into the estuary, more food source, or particularly if you have barnacles that are attaching to the seagrass blades. So this is what that macroalgae looked like on the eastern side of Charlotte Harbor. And here you're seeing this circle is where it was in the spring sampling. This is the collection that the volunteers, one sampling, one group of volunteers collected and brought back to me. And then this is from July. It's what it looked like underwater with a GoPro. And this is the footprint from July. And this actually expanded to where in January it was 15 miles from Punta Gorda all the way down to Matlacha Pass. So what caused this bloom? And is it episodic or is it long-term? Those are kind of the questions now. Some theories that we've been bouncing around. One is Hurricane Irma, which came up to the east of Charlotte Harbor. But for those people who could actually see the harbor as it was coming, we know that it blew all the water out of Charlotte Harbor. And then as the water was coming back in, because we had a lot of rain, we had a lot of flood water coming into the harbor. So did that, stir up the sediments? Did it increase the nutrients in the harbor? That's one theory. The other is between 2017 and 2019, we had a very significant Karenia brevis red tide bloom. And you see that here in Southwest Florida. This is the Karenia brevis bloom from one day in September 2018. Karenia brevis does have a low salinity threshold. So while it made it into the harbor, it didn't make it to our study site. However, it caused significant mortality of fish, marine mammals, and sea turtles. And a lot of those came into the harbor where they would have come up this eastern side and decomposed. So did those contribute nutrients? We don't know. We have a lot more questions than answers at this point. Moving forward, 2020 was a bust year for us. COVID shut us down. We weren't able to get the volunteers out in the water because we couldn't social distance safely. And so we don't have that data, but we are geared up for 2021 to both expand to Lemon Bay and add sediment samples, which Ashley is going to be analyzing for nutrient composition. And we also are geared up to be able to send some algae samples to Dale Laughinghouse, who will be able to do some nutrient composition of the actual macroalgae sample if we see these types of scenarios moving forward. Some important linkages. I hate to call them success stories when you're talking about macroalgae, but one of the things that was really important was that we were able to bring the Southwest Florida Water Management District down and show them this macroalgae because they were getting ready to release their updated swim plan, which was pretty flowery for Charlotte Harbor because nutrients were not showing up in water samples. And remember that SEAGRASS data, the aerial acreage data was showing a really nice picture of Charlotte Harbor. So when they came down, they went, oh my God, and they went back and they incorporated this concern into the updated swim plan. And then also a group of scientists that I'm working with from the National Estuary Programs and the water management districts, thinking about what we need to do moving forward. We're putting together a macroalgae workshop. So nice linkages to this citizen science project. All right, so I want to finish by taking this back to just this citizen science, because my role as an educator is to educate people and empower them. And that's how I'm evaluated. So I have 3 metrics that I use in this project to demonstrate my impact on people, and that's knowledge gain, confidence, and demonstration of skill. And so the first one, this knowledge gain, is a pre-post assessment. So coming into the volunteer training, how much they knew about seagrass and macroalgae versus after the training. And you see this 33% increase in knowledge, which is really good. The confidence is at the end of the training session, how confident they were, whether they would be able to do their survey sites. And then again, the same question at the end of July after they've done 2 sampling events. And you see this 57% increase in confidence, which was great. And then in terms of demonstration of skill, I'm using their data sheet. And if their data sheet was completely filled out and able to be used in the assessment, and 100% of the volunteer data sheets were correctly filled out, which was fantastic. So in conclusion, This project has generated really important information that was needed in Charlotte Harbor, and we think that we will continue to be able to do this moving forward, hopefully, if COVID gives us a window and allows us to do so. But more importantly, trained volunteers, and I mentioned this before, do become advocates for their estuary when they're armed with information and when they have trust in the data that they've collected. And I want to give you just one closing example, and that's from a fishing club. So I have 3 volunteers that represent their local fishing club. And this is a large fishing club. It's 500 members strong. And they came in, they weren't sure they were going to be able to do the project. They did it admirably. And this, what you're seeing here, this is a report that Ashley and I put together. We released it to the volunteers in January of this year. And in February, I got a call from my 3 volunteers saying, we are going to present this to our fishing club and we want you to come. And so I came as, you know, to be able to answer those hard questions, but I didn't present. I sat in the front row and I watched my 3 volunteers talk about the importance of healthy seagrass communities and communicating this complex ecosystem issue of eutrophication and macroalgae. And it was so impactful. And at the end of the day, I was so proud. And I'm going to end right there. And with that, I will take any questions. Thank you.
Awesome. Thank you, Betty. That is a very hopeful way to end it, and appreciate very much that idea of the citizen scientists as educators of the rest of the community. So really appreciate that. I may cancel that. So any questions for Betty? Andrew, I see that you have turned your camera on.
Yeah. Hey, Betty, that was a nice talk. Thanks for presenting to the group. And I'm curious, I'm on the faculty in environmental engineering sciences, and I haven't engaged in a lot of outreach yet, but I hope to kind of increase my presence in that realm after having been at UF here just for a couple of years. And I'm just curious. So if you can maybe expand a little bit on sort of how you find and recruit the folks that you've worked with through this citizen science project, and how much of that is sort of just engaging with people that sort of find you versus what strategies you have for going out into the community and finding the people that you think really could benefit.
Yeah, that's a great question. And I recognize your face from one of the Sea Grant Zooms.
Right, that's right. Yeah, that was just a week or 2 ago.
So yeah, I have been in Charlotte Harbor working for 20 some years. So I'm not a stranger to my community. I've been a Sea Grant agent for 16 years. A lot of it is getting out in the community and engaging, giving talks, those types of things, doing programs. I do quite a bit of citizen science. So as some of them are, you know, 1-day events, and they might be a little bit more family friendly, fun events. But from that, I keep track of everyone. So I have a nice database that I usually start out with, because these are people that I know are pretty committed to volunteering. Oftentimes they make good volunteers, but I don't limit myself. I always put out media releases. And in fact, the flyer that you saw, that did go out as a media release. And it's just, to me, it's interesting who shows up. A lot of times people aren't really sure. That's where that training comes in. If they want to do it and they're not really comfortable, I try to align them. I'll stick, you know, a county biologist on their boat, or I'll go out on their boat to make sure that they're, you know, that somebody's there to hold their hand and get them through it. And then if at the end of the day they decide that's not for them, then it's not for them. But it's really spending time and building those resources, I think, and then drawing them back in, because some people really crave that stuff. And I will mention the other part of it is not so much with this project, but with some of the other projects that I do, I can give community service learning hours, which is really important for a lot of high school students, particularly if they're going to use various grant funding and stuff. So I do tend to get a lot of younger people who are, you know, what better way to jumpstart your career and get those service learning hours snorkeling in the water versus cleaning toilets somewhere. So so there's always that avenue as well. But I will say Charlotte County is also it's the second oldest county in Florida. So we have a really healthy, retired, active population that are typically willing to come out and play with me in the water. So there's that too.
Yep, great, thank you.
John, I see that you got a question for Betty.
Hi there, Miss Betty. That was an awesome presentation. Thank you so much for taking the time out of your day to share and be a part of this. It especially spoke to me because I kind of want to be in extension as well. And so my question is, how did you get involved with IFAS extension?
Sure, that's an excellent question. So prior to IFAS, I worked for the Charlotte Harbor Aquatic Preserve, so FDEP, and I did spend some time with a nonprofit. I will say my entire career has been in Charlotte Harbor. And I always say Charlotte Harbor is kind of an academic hole. We don't have a major university that's really plugging research into our area. We're a little bit too far north for Florida Gulf Coast University, a little too far south for the University of Florida or South Florida. But so the opportunities are far and few between for a position within IFAS extension. My predecessor actually passed away, and I did know him well because it is a small scientific community that works in Charlotte Harbor. We did do some things together, and so when he passed, unfortunately, I was able to capitalize on that and interviewed, and apparently interviewed well enough to assume that position. Either that, I actually interviewed right after Hurricane Charlie, and my house still didn't have power. And so I drove to the university the day before my interview and was actually putting together my required PowerPoint presentation. So sometimes I joke about maybe I got the poor person who'd just been through a Category 4 hurricane points as well. But I think I've done well. So that's an excellent question, and I hope you do engage in extension. I think it's a fantastic career path, and I really do enjoy working with community members. It's very rewarding.
Awesome. Thank you so much for sharing again.
You're welcome.
Betty, we have a question from the YouTube from Dr. Elliot White at University of Virginia. He says, great talk. How does the scientific community at large engage with the data generated from your citizen science? And have you received any skepticism? How do you deal with any skepticism that may arise from the ivory tower side when it comes to, you know, verifiability, trustability of the data?
That's an excellent question. And yeah, I am really happy to report that the scientific community has really embraced the data that the volunteers have collected. Like I said, you know, when I presented this project at a regional summit earlier this year, I immediately had 5 texts on my phone after the talk was over. And that was what brought the scientists from the Water Management District to Charlotte Harbor to look at the seagrasses. That's how we formed this little group to plan this workshop surrounded around macroalgae. And so I think that there's a lot of interest. I mean, yes, it is not held to the same rigor as what some other scientific projects are. And I think that's why those methods are really important, and thinking about what we absolutely need to know versus what is nice to know, and focusing on the things that we absolutely need to know so that the volunteers can learn to do those well and consistently over time. But no, I haven't experienced that. Now, I do know that there are some projects, and I do compete with this a little bit, where sometimes you have citizen science projects that are kind of advertised more as like a fun family day. It's a party on the water. And so that does tend to bring the citizen science down, but I think we use enough quality assurance. You know, we bring them back together. We do the hands-on training. We have them working in groups to kind of QA, QC what they're calling 50% versus 60%. And we try to document that so that that goes with the data. So.
Yeah, I think that's great. I mean, you know, the QA, QC to have it verifiable, and then also, you know, it is the flag that goes up and say, hey, we're seeing something here. And without eyes on the SEAGRASS, you don't have anything, right?
Yeah.
You only have the data coming from the swim report. So I have a question about that, which was when you opened up, you showed the sort of bar graphs of the different aerial coverages, and you noted that you and the community, or maybe the community at least, were skeptical of those numbers. I think one thing to note is that even the variation on those areas was just really tiny. So it was like 17,000, 18,000, 19,000. The bars look very, you know, very high, or, you know, not that different. But really, if you look at it seems like the variation that they were measuring was maybe even smaller than what was represented in that, because the bars went way down to the bottom. So how do you, I mean, I guess the connection is, can you trust citizen science data? Yes, hopefully if verifiable. Can you trust, you know, agency data? And how does your community, you're like going in between in a sense, how does your community see the agency side? Sort of a long way to get to the question.
Mm-hmm.
You use your trusted, or can you, or is there a concern with using your trusted position to encourage advocacy within the groups that are working with you?
Excellent question. So as an extension agent, I don't advocate. I do try to empower people with data so that they can make their own positions. And I try to link them with those resources. So we have brought the Water Management District to, they do participate at the local level through the National Estuary Program, which does have a Citizen Science Advisory Committee. I have written articles about, you know, what their data says and what their methods are, and you know, how they go about ground truthing their methods. So we try to present all sides of the angle so that people can make their positions. But I certainly won't advocate in any direction. I often say, like, you know, we don't know. That's a great question. And but I will say, from talking to the folks that I do at the Water Management District, they're encouraged by the citizens, and rightfully so. And you know, even if, you know, we could say that they're kind of bashed a little bit in this project because of the aerial photography, they feel like that's a good thing, that people should be asking those hard questions. And so so I think that is a good point. And you know, they did just fly the 2020 aerial photography, and when they came down, they went, wonder how we're going to ground truth this.